Hi, welcome to Bio24's lecture on animal cloning. First, a brief introduction. Animal cloning is the process of making a copy of an animal cell only so that you copy the DNA. When we clone animals or plants or anything today, what we're really doing is just copying the DNA. We are not saying that we can make an exact copy of that cell, down to the molecular level of all the proteins and sugars and fats. So this is essentially like two identical twins. They have the same DNA, but some other features may allow you to tell them apart. If one twin were to dye her hair or eat a little excess food or exercise less, you might be able to tell two identical twins apart even though they have the same DNA. That's because DNA doesn't determine everything. The environment changes what you look like and what organisms look like pretty substantially. This is also true of when we clone animals. Now, cloning organisms, it turns out, is not that much of a challenge. Cloning bacteria, for instance, is exactly how bacteria divide, so there's no challenge at all. Cloning plants is fairly straightforward generally as well. Humans have been cloning plants for thousands of years. Many plants, all you have to do is take a branch, cut it off, stick that branch into the ground, and roots will grow from that branch. Then you'd cloned a tree or a bush, because the new organism, complete with roots, stems, and leaves, is an exact copy of the original adult. However, cloning animals is a little bit more of a challenge. There are some important concepts to understand when cloning animals. Animals, like plants in fact, begin when a sperm cell fertilizes an egg cell. The resulting cell is human, but it has no specific characteristics. We refer to this cell type as undifferentiated because there's nothing that makes that cell unique. It's in fact like a stem cell, a very potent stem cell, because it can make any cell type of the body. We all started this way, by a single cell that when it started to divide became more and more differentiated until eventually that cell was able to form all the different cells of the body. As this initial cell, which we call a zygote, divides and forms 2 cells, 4 cells, 8 and 16, 32, and so forth, it becomes an embryo. Even embryonic cells generally can take on different cell types. In fact, this is how identical twins are formed. They all start with one fertilized egg cell, a zygote, that then divides into a cluster of stem cells, and somehow, for many different possible reasons, this cluster ends up falling into 2 pieces. And this whole group of cells can end up forming any different cell type of the body, and in fact forms an entire fetus and baby. And these cluster of cells, again, can form any cell of the body and form an entire fetus or baby. So that process of taking these undifferentiated stem cells and creating all these different cell types is called differentiation. Those cells, once they formed these different cell types in a baby, and of course as also an adult, are then what we refer to as differentiated cells. Now, the trick to cloning is to take a differentiated cell, like say a liver cell or a muscle cell, and have the process go in reverse so that it loses its identity. That process is what's known as de-differentiation. So with cloning, we'd like to de-differentiate the cell, removes the cell's identity, and then get the process to go forward again so that this cluster of cells can form all these different cell types. So it's not that a muscle cell forms a blood cell. A muscle cell dedifferentiates into a stem cell, and then the stem cell forms these different cell types. Now, to remove a cell's identity in an animal cell was quite a challenge. It was only after a lot of research and experimentation did scientists discover that the way to do this was to place cells under starvation conditions. And in doing so, essentially turn the cell into a dormant, or what's technically referred to as a quiescent state. Once these cells are turned into a dormant state, they essentially forget what kind of cells they are, and then can form into lots of different types of cells, much like a stem cell would. Of course, forming those many different cell types and differentiating again requires reactivation of the dormant cell. The very first animal cell that was cloned was Dolly the sheep. This is how the cloning of Dolly the sheep occurred in 1996-1997. What was done was this sheep, a Finn Dorset ewe, was cloned. Specifically, cells from her mammary glands were harvested. And these cells were starved of nutrients and put into a quiescent state. They lost their individual characteristics and properties and dedifferentiated in this environment. Now, this is the cell that you want to clone, but you have to activate this cell in order to get that cell to imagine or feel like it is a fertilized egg. The best way to do this, they found, was to fuse this quiescent cell to an egg cell. And that way, that resulting fused cell could pretend and act like it's a fertilized egg cell. So you would need to harvest an egg cell from a donor ewe. The problem with this, of course, is that every egg cell contains a nucleus with half the DNA of the parent. So if you directly fused the donor mammary cell from the sheep you would like to clone with the donated egg cell, you would have 1.5 full copies of DNA. And that's too many. We just need 1 full set, not 1.5 full sets. So you would need to eliminate the nucleus from that unfertilized egg cell you've harvested from the donor ewe. And that's exactly what the scientists did, using a tiny syringe, referred to as a micropipette. They essentially sucked out that nucleus. This egg cell is then referred to as an enucleated egg cell because it no longer has a nucleus in it. However, it does still have all of the proteins, all of the sugars and fats, and all of the ready mechanisms to be a fertilized egg cell. It just doesn't have a nucleus anymore. And where does it get the nucleus? It gets the nucleus from that donated cell from the sheep we'd like to clone. So you take the enucleated egg cell and the donor mammary cell, and you fuse them. And the way that these cells are fused, in fact, is by a small electrical pulse. It's like an electrical charge. And that causes the cell membranes to fuse and the contents of those two cells to mix. What's interesting is that this, in fact, mimics the process of when a sperm cell hits an egg cell. Because one of the more beautiful things I've seen in the developmental process of human embryos, and really any animal embryo, is that as soon as a sperm cell hits an egg cell, there is an electrical pulse that's shed all throughout that entire egg cell. It's almost like the spark of life that hits those two cells as they end up fusing together. And that electrical pulse that's shocked in a fertilized egg cell is mimicked by what happens in this process here. So that electrical pulse ends up stimulating fertilization in that egg cell, and the egg cell begins to go through cell division, just like it would if this were a fertilized egg cell made naturally. Eventually, this cluster of cells forms, and much like we would have with in vitro fertilization of human embryos, this cluster of embryonic cells that formed in a petri plate is then implanted into a surrogate mother. The surrogate mother that they used for this process was a Scottish black-faced ewe, and this was to make sure that the offspring or progeny, the little baby sheep, didn't look anything like the surrogate mother and was an exact clone of the donor mother, the Finn Dorset ewe. And that's in fact exactly what they saw. Now, Dolly was the first one to be cloned this way. There have been many organisms cloned since then. But after Dolly was cloned, there was a lot of investigation and research into figuring out if Dolly was going to be healthy, if there was any issues with her health as she progressed through adulthood. Initially, there was controversy over how old Dolly really was. Because she was a product of a donated mammary cell from an adult, was she a baby or was she an adult? Now, of course, Dolly was born as a baby sheep, so she looked like a baby sheep, just like any other baby sheep, and she grew up just like any other sheep would. So the question wasn't anatomically was she an adult, but at the cellular level, how old were her cells? Now, scientists have discovered that, in fact, cells do age. The way cells age is by the ends of the DNA inside the cell. The ends of the DNA of all of your chromosomes in your cells are capped with sequences of DNA called telomeres. Telomeres are part of the DNA, but it's essentially junk DNA or unimportant DNA. Doesn't contain any genes. It's just repetitive sequences at the very end of all of your chromosomes. Now, what scientists have discovered is that over time, as your cells divide, since the time that you were a fertilized egg until now, and this will continue until death, your telomeres get shorter. And as they get shorter, they can determine and dictate how old the cells are. Now, eventually, they can get so short as to disappear, and then your important DNA with your genes in it starts eroding as well. And this can spell death to the cell. So when scientists were asking how old Dolly was, they weren't asking, was she born as a baby? They were asking, how old were her cells and how long were her telomeres? Analysis of Dolly's telomeres indicated that, yes, in fact, her telomeres did appear unusually short for a sheep her age, indicating that cloning can eventually lead to aging of cells prematurely and possibly an early death for clones. Now we're going to talk about Dolly's other health issues and Dolly's eventual death, as well as other cloning phenomena in the next lecture.